The outlines of this particular course are listed as follow. I will teach you introduction to bioinformatics, the basis. Also, I will teach you introduction to molecular biology, giving you the relevant background. And also, I will talk briefly on Sanger DNA sequencing, which is a combination of PCR and electrophoresis. Also, I will introduce you to database and tools that are used in the bioinformatics. Also, I will talk about a sequence alignment, pairwise sequence alignment, BLAST, multiple sequence alignment, protein structure and function, molecular phylogenetics, and then I have listed some reading books which are very essential for your development. And also I will show you samples of a research publication that has been done. And also, if you have question and answer at the end of the course, you can send it directly to my email address. Introduction to bioinformatics. What is bioinformatics? Looking at the word bioinformatics, we can divide it into three segments: bio, inform, and then tics. Bio, informa, and tics. Bio deals with the living thing. Informa deals with the information that we obtain from the living things, while the tics involve the techniques that are involved in assessing those information that are present in the living things. According to Lubosco et al., it defines bioinformatics as the conceptualizing biology in terms of molecules. And then we apply the techniques of informatics, and then we understand how these informations are organized on the large scale. And also Bewaji 2003 defined bioinformatics as application of information technology to the domain of biology. And the goal, which involve getting the sequence available globally and also making sense even of those sequences in order to have meaningful biological interpretation. Yes, bioinformatics is a very wide a field that encompasses some other fields, because you cannot just start bioinformatics on its own without linking to some disciplines that involve computer science and there as well as molecular biology. These are the two fundamental fields that make up bioinformatics. And you can see even on the screen, talk about computer science, talk about the hardware and the software aspects, and then the information technology, that is the technology of transmitting the information from one location and make it accessible to another location. And also the molecular biology is the one that generates the DNA even sequence, the RNA sequence, the biological information. So we obtain that from the wet lab by the help of molecular biology techniques, talking about the nucleic acid and the protein. And then all the sequence informations are available, are deposited into different databases created by the computer scientist, and then anyone can access it globally. Okay, talking about bioinformatics, as I have said, is an interdisciplinary course which involve molecular biology, biochemistry, microbiology, system biology, chemistry, the computer scientist, the computer science, and also mathematics. And also we have biomedical engineering as well as biophysics. Then we can also talk about biostatistics. So bioinformatics encompasses all the fields involved in the study of living things, as well as also some aspects of physical sciences. As I have said, the chemistry, computer science are very essential. Then statistics, to make the significance meaning of all the data that we obtain from the database. Then we need the work of biostatistics and also the help of a physicist. Central paradigm of bioinformatics involve accessing the genetic information, turn it even to molecular structure, and also from the molecular structure, we obtain functional the biochemical function of those molecular structures. And also from the function, we can determine the phenotype. Structure determines function, and function also can lead to particular phenotypes or a particular symptoms. In molecular biology, there is what we call central dogma or central paradigm. This involves the genetic information that are present even in the DNA, coded or embedded even in the nucleus of a cell. The DNA information, the genes, has to transcribe to RNA, specifically the messenger RNA, through the process referred to as a transcription process. And then during that particular period, RNA polymerase is the key enzyme that's involved in the transcription process. There are other enzymes, but RNA polymerase 2 is the major enzyme in that particular step, and it occurs inside the nucleus. The mRNA produced inside the nucleus can also be translated even to different polypeptide. It has to move from the nucleus through the cytoplasm to the ribosome, and then the protein synthesis occur at the ribosome within the cell, using the genetic code as a fundamental tools by which the tRNA determines specific amino acid that corresponds even to the codon. And the protein or the polypeptide also can give expression to specific phenotypes. Also, mRNA directly could be isolated even from living thing, and then converted even to DNA, which we refer to as cDNA, complementary DNA, through a process referred to as a reverse transcription. The key enzyme involved in that particular step is called reverse transcriptase. Also, within the cell, the DNA itself could undergo what we call replication, that is, production of copies of DNA. So that replication process, the key enzyme that's involved in that is a DNA polymerase. So this is the central dogma of molecular biology. Our information in the DNA is transcribed into mRNA, which is the one that is carrying even the real genetic information. And also from the mRNA, it translates even to specific set of a polypeptide. And that occurs even at the ribosome, as I have said, where the ribosomal RNA read even the mRNA script, and then the corresponding codon, which is the triplet nucleoside that code for specific amino acid, the tRNA sends down through the anticodon and then go and fetch the corresponding amino acid, and the corresponding amino acid are linked together by several enzymes to form even what we call a peptide bond. And at a particular point in time, the tRNA will read the stop codon, and once the stop codon is read based on the genetic code, then the polypeptide script will stop. The synthesis of that particular polypeptide will stop, and it will migrate even to the rough endoplasmic reticulum, where the necessary post-translational modification will be done even on it, and then the structural folding and all the movement through the cytoskeleton to the real site of action within the cell will take place. Talking about molecular biology, we understand that DNA is packed within the nucleus with the help of histones, and then you can see that histones help the DNA script to pack together very well into different chromosomes, and the different chromosomes could be identified within the nucleus. As you can see, we have the DNA double helix, which is about 2 nanometers in length, and then we can see it will move, and then it will form what we call the chromatin, and then from there it will reach the presence of it will form what we call a nucleosome, which consists of 8 histone protein that wind around the DNA and wrap it in about 1.6 times times. And then the nucleosome also, with the help of histones, and also they will form what we call they will form a kind of a 30 nanometer fiber, and from there everything will be packed, and then they will form a particular chromosome. So chromosome form is a segment of the DNA chain. A particular chromosome is a segment of a DNA chain. That's why in human being we have a 22 chromosome, and then the 23rd is the chromosome that determines the gender, X, X, or XY. So the DNA sequence of woman is packed within those 23 chromosomes. So any segment of the chromosome that is having problem, it means that a particular segment of the DNA is having a problem. So if a particular segment is having a problem, it's either you identify even that chromosome that carries that particular segment, and then we deal with that, or you isolate the whole DNA and then edit the DNA. So this, on this right left hand side, is the central dogma again that interprets the central dogma. We see R there is the replication, the RTC is the reverse transcription, TC is the transcription, TL is the translation. Then the genetic code that determines a particular amino acid correspond to codon is the one presented even in this particular table. Okay, let's go about just history. Let's talk about history of a protein sequence and structure, and also the one of a DNA sequence. Specifically in 1955, Sanger, a particular scientist, was the first to sequence bovine insulin. Okay. Then in 1970, Smith-Waterman, two scientists, developed algorithm that can use even to align sequence of proteins. Then in 1973, the PDB, that is Protein Data Bank, was developed and launched. Then in 1990, BLAST, which is a basic local alignment search tool, was also developed. Then in 1994, BLOCS database, which is a database that one can determine the particular motif present in a protein or protein family, was also developed. Then CAPS, which is a kind of a site or a database for assessment of protein, for classification of proteins, was also developed. Then proteomics, which is a kind of a omics technology that involves the study of total proteins present in a specific organism at a particular point in time. So the techniques that involved for proteomics was also developed in 1997, and then from 1997 to 2017, the world goes there was development in the process by which proteomics could be derived even from the genomics, by which the expression of a particular gene could be used even to predict even the protein expression. So the technology involved in that was also developed in 2017. About the DNA history, in 1953, you could remember Watson and Crick developed the DNA structure. Watson and Crick developed the DNA structure. And then in 1972, recombinant DNA technology was developed. That is, you can clone a particular DNA using a particular vector and or plasmid or bacteriophage. So you clone it using your restriction enzyme to cut even the segment of that particular vector or plasmid, and then insert your own new DNA into it, and then ligate it together by the help of a DNA ligase, and then you infect even a particular host with that particular modified plasmid, and then you culture that particular host, specifically either yeast or E. coli. You culture it, and then you test for the one that has successfully even obtained even the recombinant DNA and has your own trait of interest in it. And then in 1977, Sanger also developed technology for sequencing DNA, and that served even as a revolution even in the field of genomics. And also in 1985, PCR technology was developed. And then in 1988, NCBI, National Center for Biotechnology Information, the database, the site present in the United States of America, was also launched. And then also BLAST was also developed in that same year, 1990. As you can see, BLAST present in protein sequence structure and weight. It was also present in DNA sequence. And so BLAST, we have variants, the one that you can use to search even for protein, and then the one you can use, the variant one that you can use to search for protein or for DNA sequence. Talking about Sanger sequencing, it involves a kind of a modified PCR, polymerase chain reaction, and then the electrophoresis. You talk about polymerase chain reaction, it's a process by which you want to hasten the replication of DNA in a test tube. Let me put it like that. You want to hasten the process of replication, DNA replication within a test tube. That is, normally the DNA replicates even by itself. Maybe within the living cell, the DNA replicates. But you want to hasten this natural process so that you can obtain multiple copies of identical DNA. You want to obtain multiple copies of identical DNA, or you are interested in a particular gene segment. You want to have copies of that particular DNA segment. Then you have to make use of a PCR. But Sanger DNA sequencing make use of a modified modified PCR. That is, he inserted what we call dideoxynucleotide into each of the test tube, in addition to the normal materials necessary for PCR, which involve your normal nucleotide, that is ATP, CTP, TTP, and then GTP, that is adenine, guanine, guanidine, thymidine, cytidine. So all the 4 necessary nucleotide bases for DNA were present, in addition to the dideoxynucleotide. Then we have the forward and then the backward primer, forward and the reverse primer, and then we have the DNA polymerase enzyme present, which will help to populate even the growing chain of a DNA. And then after that he has done this process, the process is very unique, it's very straightforward. Anytime that the DNA polymerase inserted the dideoxynucleotide, the growing DNA chain will stop. And then making use of principle of PCR. PCR involves 3 steps. Number 1 is the denaturation, by which the thermocycler, which is the machine, subjected even the double-strand DNA helix to about 94 degrees C, during which the hydrogen bond that links the double helix together unwind and break, and then the DNA double helix form two strands, which serve as a template on which the primers, the reverse and the forward primer, bind onto. Then the second process is the annealing, which is the point at which the temperature drops to about 50 degrees C, during which the primer is able to form hydrogen bond even with the template. Both the template, we have 2 templates now, the forward 5 to 3 prime, and also the one that's 5 to 3 prime reverse. So the primer, the forward primer binds to 5 to 3 prime, then the reverse primer also binds to 5 to 3 prime direction. So that is annealing, and also the temperature, the machine thermocycler now increase the temperature to about 74 degrees C, which is the optimal temperature for the Thermophila aquaticus DNA polymerase. Thermophila aquaticus is a bacteria that is a thermophile that can still be active at around 70 to 90 degrees C. So the DNA polymerase obtained from that particular bacteria is able to withstand high temperature and has its optimum around 70 to 75 degrees C. So once the thermocycler increase, the temperature increase at the third step, which is what you call elongation now. At the third step, which is elongation, the temperature increased to about 74 degrees C, and then DNA polymerase is able to grow the chain of the templates together with the nucleotides, and then together with the primer, able to increase the chain. So the cycle by which you need to grow the chain depend on the length of the DNA that you want to copy. Length of DNA you want to copy. So for you to have meaningful length of a particular gene of your interest, there is a need for you to repeat the cycle so that you have enough amount, enough quantity of the DNA of your interest or the gene of your interest being amplified, being copied in a sufficient manner. So about 20 to 30 cycles is necessary for you to really amplify the segment of your DNA or your whole DNA. So what this means is that the extent or the length of the DNA that you want to amplify will determine the length that you will subject the naturation, the annealing, and then the elongation even too. So that was what Sanger did. And after he has done that, using the PCR, he now subjects the content of each of the test tube into electrophoresis, which is a kind of a biological technique that has been used to separate macromolecule, DNA, RNA, or protein in an electric field based on their charge and their size. So electrophoresis was done for each of the test tube that contained different the dideoxynucleotide, and then different bands of these samples were what showed even on the gel, by the use of agarose gel, because we are working with DNA or RNA, and they have a big size even than protein. So after the electrophoresis, the gel was stained with ethidium bromide so that we're able to see a particular spot that the DNA fragments are on the gel, and then the gel was visualized even under the UV. So he read even the what's it called now, the bands from the bottom, because the smaller fragments will be the one that will migrate faster in the electrophoresis gel. So he read even the chain from the sequence from the bottom upward, GTCCA, GTGCAA. So that one, he was able to discover the arrangement of nucleotide within the DNA samples within DNA sample of interest. So in these days where technology has increased and modified and built upon the Sanger DNA sequencing, and that's what we now refer to as the next generation sequencing. Next generation sequencing involves the library preparation. Number 2, cloning amplification, that is your gene of interest, segment that you are interested in, you amplify it. Then number 3 is involve cyclic array sequencing. And we have about 3 types of a cyclic array sequencing that are presently being used. Number 1 is the pyro sequencing. Number 2 is the sequencing by ligation. Then number 3 is sequencing by sequences. So we refer to the present generation of sequencing technologies which has been automated by the help of computer and sophisticated devices. We refer to it as next generation sequencing. So today, to sequence even a particular segment of DNA or the whole DNA of your interest, depending on the length, using different machines that are available, you can see if you want to sequence a sequence that is about 1.2 gig, you can between 9 to 12 hours or 7 or 24 hours, you'll be able to do that. You can see the bigger the size, then the specific machine that can handle it are also available. And there are sequencing technologies companies that are doing this even today. You can have Illumina is one of them. Oxford Nanopore is one of them. Also, Thermo Fisher Scientific is also involved in the DNA sequencing. Agene Technology, Roche, Microgen, they involved in shotgun metagenomic sequencing. Also, we have BGI Genomics and also Pacific Bioscience. So all these companies, they involve in DNA sequencing either from the microorganism, from plant source, or from any other marine source or human being. So you can even have your DNA sequence even today. What you need to do is to get part of your tissue and then send it to this company, and then they will able to sequence even your own DNA for you today. Venter was able, together with other scientists in 2000, they developed and helped to produce even the first draft copy of a human genome. In 2001, the first draft copy of human genome has been has been made available, and today it has helped even the discovery of different biomedical insights that can help even in treatment and then diagnosis of various diseases that are peculiar to human being and to other organisms. Yes, having obtained the sequence, what do you need to do with the sequence? What are the things that you can do with the sequence? How are you going to make sense of those sequence? If you say that you want to read a particular sequence that is about a 120 gig or 6,000 gig, you will not be able to read it humanly speaking by yourself. You will not be able to make sense of those sequences one by one and see areas where there are mutations, areas where there are some specific modulations. So in order to do that, that is where the computer comes in. And you have to understand computer in order for you to what to able to appreciate bioinformatics. So this particular section, I'll be talking about computers and the programs, computers and programs. So as an introduction to computer, what you need to know about the computer that you are working with, number one thing is you have to know the operating system. We have to know the operating. We have three major operating systems even in the market. There are so others, but the three major ones involve the Windows, which it was being developed even by Microsoft. Then we have Linux, and then we have Mac, which was being developed even by Apple. Linux, we have the different versions of Linux. We have the Ubuntu, we have the Mint, we have the Fedora, and the rest like that. But these are the three major operating systems that you can find on major computers. Then number two, you have to know about your PC. That is the specification of the device, the laptop or the computers that you are using even for your bioinformatics analysis. When you talk about specification, you have to what? You have to know the processor. You have to know the processor. Processors and the RAM, which is the random access memory, depends. The computing speed depends on the processor. That is the processor that of your computer. Then the RAM, the size of the RAM, and then we talk about the software compatibility. That is the software that you can install on your computer. It depends on your device specification and majorly depend on the operating system that you are using. You can install a software that is meant for Windows. You can install it for Linux, except there are some software that you can work. It can work on either on several operating systems. So there is no specific for the system, but there are some softwares that has to be specific for a certain operating system. Okay, so at that you have to know, okay, if you have to install Windows software, you have to install it on Windows operating system laptop or computer, or if it is a software that make use of Linux, it can only work with Linux operating system computer. Then another thing is a system type. We have the bit size, we have bit size, we have two types for now, even it has been three. We have 32 bits and then we have 64 bits. And presently, I think there is what we call maybe like 128 bits. But the most common computer in our environment, as you can see, are majorly 64 bits. There are some softwares that are produced based on 32 bits, and while some are produced based on 64 bits. A software that is a 64 bit cannot work on a laptop or a computer that is 34 bits. Okay, but the software that is produced based on 32 bits can work even on 64 bits. So that is just the case. So you have to understand all these device specifications in order for you to have a good computer that can use to analyze even your biological data. The another thing that is very paramount is the programming language. Programming language are the things that are the words or the sequence of instruction that the computer understands. They are the language that the software are built upon. The softwares are built upon, or the computer algorithm are built upon, or the computer, what's it called, a database are built upon. So there are different softwares. There are different programming language. We have Python, which is the most common one. And then we have other ones like a C++, we have Fortran, we have Java, we have Perl, we have MATLAB. And recently, even R has emerged to be one of the programming language these days. So you have to understand that, okay, if you want to develop a tool in bioinformatics, then you have to learn one or more of this programming language in order for you to write a code that will be able to work to analyze a specific biological data. Talking about database, that is where the computer science has come in. And You have to understand that, okay, database are produced by the help of the computer scientists, okay, using the available tools, okay. We have relational database and we have non-relational database. That is, depending on the information that you want to put into the database, we determine the type of database that you have to use and then the tools that you have to use to produce that particular database. We have SQL, we have Mongo, and all the rest like that. But as a biological science, or the person with a biological science background, you have to understand how database works, and then you have to understand the significance of a particular database. That is, what a particular database information that you can access from it. So this is a kind of a sample publication that you can just access from online using the DOI or the name. You can see we have application of biomolecular database in bioinformatics. There is no way you want to do bioinformatics without accessing database. Okay, you have to access a database of the existing information, the sequence, the structure that has been made available. Then you mine those database for the information that you needed even for your own analysis. So the major database in the world include the NCBI, which is the National Center for Biotechnology Information located in US. The site is there, www.ncbi.nlm.nih.gov. There another one is EBI, European Bioinformatics Institute, www.ebi.ac.uk is located in the UK. Then the third one is a Swiss Institute of Bioinformatics, and they host what we call ESPACY, which is the Expert Protein Analysis System. It is done. You can access that at www.espacy.org. Then also we have Canada Bioinformatics Resource, and also there is this particular useful directory which you can access even different softwares, different tools, and then different database directly from the index of this particular database, www.vls3d.com. Talking about database, we can classify database into 4 different categories. Number one, we have the primary database, which is where the raw sequence that is being generated from the wet lab, like the gene sequence or protein sequence or a particular protein structure, RNA structure, once it's being generated from the wet lab, it has to be deposited into what you call the primary database. For the genes, we have 3 major primary databases in the world. 3 databases in the world. We have what we call the Gene Bank, which is located in US, and also we have EMBL, which is located in UK, and also we have DDBJ, which is located in Japan. So these 3 databases are interconnected. So if you deposit your sequence in one, the other one will fetch that particular, those other ones will fetch that particular sequence. So you don't need to deposit the 3, just deposit in one. The other 2 will fetch that particular information. Then we have a protein information resource, which is where you can deposit your protein sequence even too, and also switch prot, which is also referred to as a uniprot. You can do that to deposit your protein sequence. For the protein structure, we have LCSB, also which is located in the UK, LCSB. Then we have the companion of it, even in Japan and also in UK. That one is for the protein structure. PDB structure is for protein structure. Okay. Then the second type of database is secondary database, which is database that fetch information even from the primary database. Example is a ProCite. Example of that one is ProCite. Then we have what we call composite database, which is a database that comprise of access to other database. You can access the primary database, you can access the secondary database through the composite database. Example of such is Ensemble. Ensemble, you can access information about your gene of various plants, of various organisms, of various animals. Then also you can access even their corresponding protein in an organized manner. Then lastly, we have what we call a specialized database. This specialized database, they focus on a particular organism, a particular plant. Like rice database is focused majorly only on rice. Fly database focus on flies, insects. OMI, which is an online Mendelian inheritance in man, focus on human disease alone, inheritable human disease. So those are the information that you can find on specialized database. They focus on a particular subject. Also today, the specialized and composite or specialized database, you can also categorize special database to sequence database, structural database, enzyme database, microarray database, clinical database, pathway database. That is a Kyoto Encyclopedia of Genome and Genes, keg.jp. Yes, keg.jp, kejj.gg.jp, kegg.jp. That is a Kyoto Encyclopedia of Genes and Genome. So we have all this, then we have a database for bibliography. So that is example is a PubMed, where you can access a medical related or biological related publication. Then we have chemical database. That one is PubChem or Zinc. So the PubChem is located is indexed even in the NCBI. You can access all the chemical structures of plants, of drugs, and the likes. So those are the classifications of database which serve as a basis for any bioinformatics analysis. You have to make use of the existing information about your gene, about your RNA, about your protein, about a particular chemical compound that has been made available, through which you can develop an idea and also develop an insight to analyze the existing or current or new problem. Now, this is how the expertise look like. You can access different protein results. The tools, the software you can access through expertise. And also UniProt. UniProt is for protein sequence. The protein sequence that has been made available, you will find it even on UniProt, www.uniprot.org. So once you have searched for that particular name, it will bring the result for you that has all the organisms that possess that particular protein. So you just click the entry name of your interest, of that particular organism of your interest. Just click on the entry name. So once you click on the entry name, it will take you to another page where you have the full information about that particular protein. On the page, you can have information about the function of that particular protein, the subcellular location, that is a location within the cell. If it's cytoplasm, if it's inside the mitochondria, if it's inside the Golgi apparatus, you're able to know the location of that particular protein within the cell. Then if there's any pathology, that is disease associated with that particular protein, you can see it from the UniProt. And also any other interaction with other proteins, you can see it. Then the structure, you can access the structure of that particular protein from the UniProt. It can either take you even to the protein database, or you download it directly even on the UniProt. Even the AlphaFold, which is a kind of a predicted structure of protein developed in conjunction with Google AlphaFold. So we can see that even if the wet lab structure is not available for that particular protein, a predicted structure of AlphaFold structure of that particular protein will be made available for you to work to do your analysis. Then also we have the sequence. You can download the sequence of that particular protein. Then any existing publication that support that particular protein are also available for you to access on UniProt. Yes, sequence alignment. When you have been able to download DNA sequence, RNA sequence, or a particular protein sequence, you have to what? You have to make sense of those sequences. You have to derive biological meaning from those sequences. Any sequence that you obtain from DNA or RNA from plants, DNA, RNA from a microorganism, DNA, RNA from any animal living thing, then you have to what? You have to align, compare your sequence with others, and then see the area of similarities and area of differences. So sequence alignment is widely used in bioinformatics. It's very central to bioinformatics. And then you can have the proteins or your gene, which are various of different lengths, and then you have to what from different species, so you can see how one species differs even from another. So it can also help us to determine the types of mutation that occur within the same species or within or across even the species during the course of evolution. So why do we need to study sequence alignment? It will help us to understand the function of activity of a new gene or protein. You just stumble on a particular new protein that you don't know the function. So by comparing it with the existing protein, you'll be able to see, okay, if they are similar, then based on their similarity in structure, then you can what? You can deduce even the possible function of that particular protein. Then you can also tell us about the structure or shape of a new protein, the location within the cell, the location of that particular protein within the cell, the stability of that particular protein. It will help us. Then the origin of that particular protein, you'll be able to pinpoint the origin where that particular protein is coming from within the cell or within the what's it called, the phylogeny and times of evolution. Then also we talk so many others. So sequence alignment is very, very paramount, even when you are doing drug discovery and you want to see the function of a particular drug. So you can align several organisms and see which of this organism, which of the protein of this organism respond even better even to your particular drug. So we have two major types of sequence alignment. We have the pairwise sequence alignment. That is pair. You have two sequence only. You want to compare two sequence. Okay, sequence A, sequence B. You want to align them. Then we have multiple sequence alignment when you have more than two sequence. We call it multiple sequence alignment. You want to align them to see area of their match, area of their mismatch, and the rest like that. So sequence alignment is very, very important. Okay. If you talk about sequence alignment, there are two terms that are very essential. Number one, we have what you call sequence identity, which refers to it as the percentage of match of the same amino acid residue between two aligned sequence. We talk about match. That is, you have alanine in the first one, you have alanine in the second one. So that is match. It belongs to the same set, is the same set of amino acid. You have A, you have it also in B. Then if you talk about similarity, it refers to the percentage of aligned residue that have similar physicochemical properties, characteristics, and can readily substitute for each other. You have for example, let's say now you have a positive charge, let's say like lysine, even in the sequence A, and then you have let's say now like arginine in the sequence B. Yes, they are positive charge amino acid. So they can still be found even in the same area within the protein. Not that you have a negative charge in the first protein and then you have the positive charge in the second protein. They are not, in terms of physicochemical property, they are not similar. So when you have amino acid in A that belongs to the same, let's say polar sequence in A, then you have another polar amino acid in B. Though they are not the same identical amino acid, but they are polar amino acid, then the percentage similarity could be deduced. These are the formula for you to determine similarity. S, when you have the length of the what's it called, the LS times 2 divided by LA plus LB times 100. S there is the percentage of sequence similarity. LA is the number of aligned residue with similar characteristics. Number of aligned residue with similar characteristics. Then LA and LB are the total length of the protein of each individual sequence. The length of the protein A, let's say is 50, then the length of protein B, let's say is 40. So when you add it together, which is 90, then you have the sequence that align in terms of similarity and identity. You count all those ones, then you divide by the total length of the two, then multiply by 100. Then for the identity, it can be calculated in similar manner like similarity. You look for the amino acid residue in protein sequence A and protein sequence B that are identical. Alanine, glycine, phenylalanine. So those are the ones that are identical. But for the similarity, you can have alanine in one, then you have glycine in the second one. Yes, they are still a kind of aliphatic aliphatic protein, aliphatic amino acid, they can what? They can still go for each other. So you count that one based on the number of similarity. So for the identical, you have the same amino acid in the first protein, the same amino acid you have in the second protein. So you count the number, you multiply by 2, then you divide by the total number or total length of the total amino acid present in the first protein and the second protein, and then you multiply it by 100%. So the other formula for you to calculate even the identity and then the similarity. So talking about pairwise alignments, you have to understand that proteins are very versatile than nucleic acid. Why? Because nucleic acid are made up of only 4-letter alphabets. Only 4-letter alphabets. But for proteins, you have at least 20 natural amino acids. 20 natural amino acids that has to what? That has to undergo combination in order for that particular protein to align, or it has to undergo what we call a kind of shuffling in order for that particular protein to align. So proteins are very versatile than the nucleic acid. And for you to do pairwise alignment, you want to compare two proteins sequenced together. We have two major methods. Number one, we have what we call the dot matrix. That is, you write the sequence of the first protein on y-axis at the top, and on the y-axis, then you write the sequence of the second protein even on the x-axis at the top. Then you align each one by one, each one by one, each one by one. So areas where we have a kind of the same set of amino acids, you put dots. So you dot, 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 dot. Then after that, we now draw a line to show the area that match, So based on that, you'll be able to what? You'll be able to develop a kind of a similarity pattern for the two proteins. So we have some tools that you can use to do that. You don't have to do it even manually. Some of the tools include dot matcher, dot helix, and then matrix plot. The second method for doing pairwise alignments involve what we call dynamic programming method, DPM. Dynamic programming method. It's a method that determines optimal alignments by matching two sequences for all possible pairs of characters between the two sequences. That is, it doesn't just do a kind of okay, this one match and put dots. It finds different combinations. Let me put it like that. Different combinations of aligning the two sequences and then determine the one that has the optimum positive value. So the example is indicated even on this particular slide. Let's say you have a sequence that's made up of let's make all of this particular sequence 1 ACA, then sequence 2 GACAT. You know, this particular sequence could either mean that your DNA, or it could either mean your protein, because the ACA there could either be your adenine or your cytidine, while it could also be alanine or your cysteine or your threonine. So either way, these are just a sample of a sequence that we want to align. Then for dynamic programming, there is an introduction of what we call a scoring guide. If a particular amino acid or a particular DNA sequence in the sequence 1 match the one in sequence 2 during the alignment, then we have a kind of a score. For match, we have 8. For mismatch, we have 2. For gap, any gap introduced during the alignment, we give it -5. So you can see the optimum alignment that dynamic program we try to find. We have 1, 2, 3, 4. So we try to align each of these sequence based on their match, mismatch, and also introduction of gap. So looking at the first one, you have the score where you have A at the first sequence and then you have G at the second sequence. It's a kind of mismatch. So it has a score of 2. C and A is a mismatch, it has a score of 2. A and C is a mismatch, it has a score of 2. Gap and A is a mismatch, it has a score of, it's a kind of a gap, so it has a score of -5. Then we have a gap again, That's a score of a -5. A score of -5. So as a sort of that, when you combine that two together, you have a score of -2, a score of -2, a score of -2. So there's a kind of a linkage over there. So if looking at that one again, for the second one, you have a gap, and then that one will be -5. That's where you see, that's what you see there. Then the second one, AA, that one is a match, you have a score of 8. The second one, the third one, CC, that is a match, you have a score of 8. The third one, AA, that is a match, you have a score of 8. Then a gap, that is score -5. So summing everything together, you have a score of 14. For the third one, you have a gap, which is a score of -5. Then you have a match, kind of a match. Okay, you have another gap. You cannot see. Let me expand this. You have another gap, so that's another score of -5. Then you have a kind of a mismatch. You have a kind of mismatch, which is AC. That is a +2. Then you have another gap, which is kind of a CA. You have that is another +2. Then you have another mismatch. You have another mismatch, which is 80. That's a kind of a +2. Summing together, you have -4. Then the last one, we have AG, which is kind of a mismatch. The other one is 2 + 2. Then you have two gaps. That is -5, -5 down below. Then at the top again, you have a kind of a minus. You have a kind of a dash, dash, dash. So as a sort of that, which are not identical, then you have -5, -5, -5, -5. Then total of that one is -28. So looking at this particular 4, you can see 14 is the one that has the optimum positive value. So that is the one that the dynamic program will pick for you as the best alignment, which is the second alignment over here. Second alignment. So the one with positive value +14 is the one that has the optimum alignment, optimum score. So that is how dynamic program works. So in the alignment, this is a kind of a real analysis of two samples. In the alignments, areas where you have okay, there's a kind of this particular one, but let me just make mention of it. The arrow is not pointing to the right thing. So area where you have a line between the top and the down, that is the area where you have identity. Look at the first one here. Based on that, it's supposed to align with M, and M supposed to have a straight line. K and K supposed to have a straight line. Okay, so that is about identity, where you have a straight line between the first and the second. That is identity. But where you have just one dot, one dot, that's a kind of somewhat similar. They are not really similar, but they can still kind of substitute for each other. Area where you have two dots, that is the area where they are very similar. Okay, they are very similar. Then there's an introduction of internal gap. Okay, internal gap has a lesser score than what is it called? We have internal gap which have a score. Then terminal gap also has a score. Okay, internal gap has, I think it has higher score, that is more negativity than the terminal gap. Okay, so all the dot dash 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 dash, those are the internal gap. Then if the dash 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 at the end of the sequence, occur at the end of the sequence, then we call that one terminal gap. Okay. Then in a sequence alignment, we have two major classification. Yes, we have two major classification. We have the global alignment, and then we have the local alignment. Global alignment, when you have two sequence that are potentially equivalent. That is, even looking at the sequence, you can see that they are not too diverse. They are not too varied from each other. Okay, so the goal of the global alignment is for you to identify the conserved regions and then the differences. Okay, and one of the key algorithm that is being used for global alignment is a Needleman-Wunsch dynamic programming algorithm. Dynamic group program algorithm. Okay, when you want to compare two genes or two proteins of the same function, but it could be from different species. It could be from, we should know that their similarities will be very high, while their differences will be somehow small. Okay, so the alignment that you can use for that is a global alignment to give you a good score. Then for the local alignment, when you have two sequences that may not be related, okay, then you have to use local alignment. Excuse me. Yes, then the algorithm that is being developed for that I can use for that is called Smith-Waterman dynamic programming algorithm. Maybe you just sequence a new gene, okay, and you want to identify that particular gene. You want to what? You want to match it with another set of sequence, okay. So you have to use local alignment. So that particular one will look for local matching and then do the alignment based on that particular area. Let's say you have a particular microorganism, you have done the 16S RNA sequence of that particular microorganism, and you now want to what? You want to align that particular your new sequence from your micro. You want to align it even with another sequence. So what you should look for is just the area of conservation. Then you make use of a local alignment. We look for the conserved domain or the motif that are present within the two aligned sequence. Then this is a kind of example or the overview of a global and local alignments. As I have said, Needleman-Wunsch is the one that was developed even for global alignment. It compared the length against length. Protein A from the length 1 to the end, it compared with protein B from the first to the end sequence. But for the local alignment, which make use of a Smith-Waterman algorithm, it considered only the area that are somewhat similar. So it will look at the conserve or the area of similarity in the protein A and then compare it with the protein B or the second sequence, the gene A together with gene B. So they consider that area of similarity, as you can see in the figure presented on this particular slide. Then another alignment method that has been developed is what we call heuristic alignment. Heuristic alignment is for the pairwise sequence alignment also, where it search or you align the proteins or the DNA, looking at the protein 1 against protein 2. And this one find expression when you are searching against large database. You have your sequence available and you want to align it with each of the sequence present in the database. Your own protein, which is protein A, you want to match it with different proteins that are present in the database. This particular algorithm, BLAST and FASTA, which I will explain in the course of this particular study, we take even your sequence and match it one by one with the sequences that are present in the database, and then give you their score, their percentage identity, and then their percentage similarity, the Z-score, and the E-value based on the extent of the information or the proteins that are present in the database that you are using. Okay, I will come back to BLAST in the course of this particular course. That's what we call a substitution matrix. Specifically, since I've told you that proteins are very diverse in nature than DNA, so we have substitution matrix for protein, and also we have for DNA, but the most common one is for the protein. Okay, because there are different mutations that can easily be evident even in the protein. So this particular substitution matrix can look at it closely to determine the substitution rate, that is the rate at which mutation occur within a particular sequence. Okay, there are two most commonly used models. We have the blossom and then we have the palm. I will tell you the full name now. We have the blossom and then we have the palm. If you talk about blossom, that is a block of amino acid substitution matrix. Blocks of amino acid substitution matrix. It can align conserved regions of evolutionary divergent sequence, cluster at a given percentage identity. It can their frequency of amino acid and the rate at which the substitution takes place. And we have different variants of of a blossom that has been developed. Okay, we have blossom 45, blossom 62, blossom 90. So the higher the blossom number, the more closely related. That is, blossom 62 is closely related to blossom 90. Okay, and depending on the sequence that you want to align, we determine the kind of a blossom that we have to choose. But looking on the average, blossom 62 is the one that is at the middle. But if you want to match the proteins that are somewhat different even from each other, then you have to what? You have to use the lesser blossom, that is blossom 45. Okay, but if you want to use, if you want to consider like what's it called now, protein that are almost similar species, that is the divergence is as less, very less, then you use a blossom 90. For also PAM, which is point accepted mutation matrix, point accepted mutation, it help us to observe mutations in a set of closely related protein. It makes use of a Markov chain model, that is a kind of a neural network, to determine the kind of substitution that takes place. Okay, so it normalizes so that PM1 is equals to 1 mutation per 100 of amino acid. That is how it works. 1 mutation per 100, and then the PM1 will be the one even that can help us even to determine that. But when you have multiple mutations in the set of sequence that you are considering, then you have to use what you call the I number, PAM number. When the mutations or the substitution or the differences between your protein are very wide, are very enormous, then you have to use higher PAM number kind of matrix that we are going to use. So looking at the table even below here, you can see PAM number 1, observed mutation just 1. But look at the PAM 250. PAM number 250, the observed mutation that one can accommodate is about 80% mutation rate. 80% mutation rate. So you can see the identity will be what? Will be reduced drastically as the PAM number is increasing. So it's a kind of a opposite of a blossom. For the blossom, as the blossom number is increasing, then it's based on less divergence. But in the other way around, as the PAM number is increasing, it is to the more divergent. Take note of that. To the more divergent. Yes, we have to make sense of interpretation or the sequence alignment. We have to understand what are the insights that we derive from our sequence alignments. Number one, we have what we call homology. So when it's a kind of a similarity that you observe when you align your sequence, which can help you to inform whether they come from the same common ancestor. So based on the similarity, the pattern similarities and the identity and all the rest like that. So when you compare the two, and you can see that they are somewhat closely together. You have a kind of a score that is above, let's say 50 above, then you can say they are homolog. But you can still define them based on their identity, particular identity, and their sequence identity. So we have two types of homology. We have the autolog. When you have homolog sequence from different species that arose from common ancestral gene during speciation. Species A and species B, you have the same protein performing the same function, but they differ in one small sequence or the other. Okay, so they may or may not be responsible for a similar function. Okay, the differences is due to speciation events during the course of evolution. One species emerge from the father, another one emerge from the father. So but they go different direction. So a particular parent give birth to two children, okay, and two children now, they go into their different location, they go into their different location. As a result of that, their offspring, they were what? They adapted even to the environment that they find themselves. As a result of that, the way their behavior, the way their function, everything may no longer be the same. May no longer be the same. So that is about ontology, orthologs. Therefore, the paralog, when homologous sequence within a species, a single species that arose by gene duplication. Within a species, you have two sets of protein performing the same function, okay? And sometimes they will not perform the same function, okay? So when you have paralog, you have two sets of protein within a species performing usually different function. Protein A, protein B in a species, but they come from the same, or they have the same kind of similarity, but their functions are not the same. They present in different tissue, okay, and the function they carry out over there depending on what is going on within that particular tissue. So similarity give birth to homology, So the number, the percentage of similarity and identity give birth to homology. So similarity versus homology. Similarity refers to the likeness or the percentage identity of two sequences, as I have said, and it has to be statistically validated. Statistically validated. You have the p-value, you have the e-value, you have the z-score. Okay, so by doing that, you're able to define the similarity. So similarity does not imply homology. Okay, does not imply homology. Okay, they may be similar in one way or the other, but once the similarity threshold is not established, then you cannot define the homology. But for the homology, it refers to a shared ancestry when they emerge from the same ancestral establishment, ancestral factor, ancestral organism or ancestral protein or ancestral gene. Then you can say they are homologous. When two sequences are homologous, they are homologous if they are derived from a common ancestral sequence. Homology usually implies similarity. Yes, homology usually implies similarity. Okay, so just like we say that all enzymes are protein, but all proteins are not enzymes. So the same thing applies here. Homology is based on similarity, but similarity does not imply homology. Similarity does not imply homology. For us to establish that two proteins are homolog, okay, there are criteria that we have to establish for the two proteins. There are 6 criteria listed there. Number 1, the DNA sequence of their genes must be similar. The DNA sequence of their gene must be similar. Number 2, the amino acid sequence also must be similar. Number 3, their 3-dimensional structures must be similar. That is, protein A, protein B, their structures in terms of the configuration of the structure and the conformation, it has to be similar. Then number 4, their enzyme substrate interaction are similar. If it's an enzyme, okay, the way they catalyze the substrate must be the same. Okay, then number 5, the catalytic mechanism must be similar. That is, if it is using metal-ion catalysis, the second one must be able to use metal-ion catalysis. If it's an acid-base, general acid or general base catalysis, then the second one must make use of a general acid, general base catalysis. If it's a covalent catalysis, the second one, the first and the second, they must behave making use of covalent catalysis. Then lastly, number 6, the segment of the polypeptide chain essential for catalysis are in the same sequence. If it is present on the C terminal, then in protein A and protein B, it must be present in C terminal. If it's present on the N terminal, then in protein A and protein B, the segment that catalyze the essential that involve essential catalysis must be similar. So these criteria are in descending order of strength. If criteria 1 and 2 holds, then the rest will follow in most but not in all the cases. Why? Because if you have a DNA sequence A and DNA sequence B, by the time they undergo the open reading frame and then the conversion through the, what's it called now, the genetic code, there is possibility that two DNA sequence can give birth to one particular protein amino acid protein sequence because of the degenerate of genetic code. Okay, because of the degenerate of genetic code, when you have two or more codon coding for a particular amino acid. Then you have to note that in serine proteins, which are an example of a particular enzyme, their tertiary structure are more concerned than primary structure. The primary structure, okay? When you are looking at the sequence, that is the primary structure of that particular protein. But when you are going from the primary to the secondary and to the tertiary, okay, the definitely of the similarity can now be further be established. Can now be further be established. So function, can change even based on the criteria 3 or 4. Okay, criteria 3 or 4 may not hold. Okay, sometimes structure has been conserved through evolution, but function has changed. They have the same structure, but the function is different. So as a result of that, you have to rule out number 3 and number 4. Okay. Yeah, this is a kind of example of where you can carry out a pairwise alignment on a European Bioinformatics Institute EBI by accessing www.ebi.ac.uk/tools. Then you can click on the what's it called now, the necessary tool that you want to use. For this particular one, you click on a Niduam Wunsch algorithm, which is part of MBOS on this particular site. So you have an area where you can choose the type of your sequence, the type of sequence that you want to input. This is a protein sequence or nucleotide sequence. So you click on the appropriate button. There you paste even your sequence into the sequence bus, and then if you want to match the two, then you put the first sequence in the first box, and then you put the second sequence in the second box. Then if you want to modulate the parameters, okay, if you are familiar with this process and you want to check or change some parameters, then you check some parameters, and then over there you click submit. So it will give you the alignment of the two sequences based on global alignment. It will Wunsch global alignment. Then another alignment, as I have talked about, as I have said during the when I mentioned heuristic algorithm, that is when you want to do pairwise alignment and you want to use your protein of interest to query large database consisting of thousands of different proteins. You want to align your own protein with those thousands. Then you need what we call heuristic algorithm. An example is FASTA, and then we have BLAST. So talking about FASTA now, which you can access on the EBI site, FASTA means a fast approximation to Smith-Waterman. What you can deduce from this is that this is a kind of local alignment, but it does the local alignment work in a faster measure, in a faster way. Okay, so FASTA can scan for protein and DNA sequence library for similar sequence. Then we have a variant. There are different variants of FASTA. We have FASTA X or Y, which compare DNA sequence to protein sequence database and comparing that to translated DNA sequence in forward or reverse translational frame, 5' to 3', 3' to 5', and then consider that particular similarity. Then we have T-FAST X or Y. So compare the protein sequence to a translated DNA sequence. So if it's for FAST-Y, that will be the other way around. You compare DNA sequence to a translated open frame protein data bank. Then we have FAST-F, which compare ordered proteins. We have what we call Edman degradation of protein. Okay, you want to see how a particular protein will degraded. So this particular FASTA-F will do that even for you. Then we have FAST-S, which compare on other protein, which is based on mass spectrometry, that is the fingerprint of the particular peptide or particular protein in form of peptide. You can predict that by using FastX. Then we have search. This is a rigorous scan of protein or DNA sequence library using Smith-Waterman algorithm. Then the second one is BLAST, which is a kind of heuristic algorithm, heuristic alignment algorithm. BLAST means basic local alignment search true. Basic local alignment search true. Okay, so instead of narrowing the dynamic programming search space, BLAST works a different way. BLAST works a different way, and you will see. First, it creates a word list. Okay, both of the exact sequence and then high scoring substitution. High scoring substitution. I've talked about the scoring guide. Match, mismatch, and gap. So BLAST does it well and it does it faster. So the BLAST also is available on EBI and also is available on NCBI. So BLAST, which is a basic local alignment search tool, we have different variants. We have different variants of BLAST. We have BLAST P, which is a protein sequence versus protein sequence library or database. You have a protein sequence, you want to query it against protein sequence database, then you use BLAST P. Then BLAST N, when you have nucleotide, your nucleotide sequence, you want to use it to query nucleotide database. That's BLAST N. You use BLAST N variant of a BLAST to do that. Then BLAST X, when you want to translated DNA, you want to use it to what? To query protein sequence. So it will first translate that DNA to mRNA. Then from mRNA, you now use it to query the protein sequence database. Then we have another BLAST, which is a WU BLAST, where you have other variants of what is it called, of BLAST. I have talked about BLAST S. Then T-BLAST N. So T-BLAST N, you use protein query. You use a protein sequence to query translated nucleotide database. So this particular one, we query the translated nucleotide using the 6 open reading frame. That is, if you check the genetic code and use all the open reading frame that are possible to deduce the right DNA sequence. I've told you a particular codon or one or more particular codon can code for a single protein, a single amino acid. So as a result of that, when you are using T-BLAST N, so that particular algorithm will consider various channels, 6 open reading frame, that you can get your corresponding DNA sequence, the optimum one. Okay. Then for the T-BLAST X, when you are using translated nucleotide query to query translated nucleotide database, okay, your own DNA, you want that particular one to first undergo the open reading frame, and then now use that one to determine or to detect or to align with other DNA, So that means your own DNA will first undergo 6 open reading frame, ORF. Then that's all those 6 open reading frame will one by one undergo 6 open reading frame against the query database. Again, that means 6 times 6 making 36. So that is how BLAST works. Then this is an example of a BLAST on the NCBI. You paste even your sequence there in faster formats. If you talk about faster formats, your sequence has to start with a greater than sign, followed by a description of that of protein or gene, then the OS, which is the organism species, then we have the GM, which is a gene name. So the greater than sign must be there. So once the algorithm read that particular greater than sign, it know that first line is for description purpose. Description for one is see that particular greater than sign, or you know that okay, that particular line is the line that describes that particular sequence. So after that line, all the sequence for your protein or for your gene will now follow. So this is how the BLAST what's it called, blast P look like. You put your query sequence there. Then you select the right database. And then if you have any organism that you want to exclude from your results, you click on exclude. Then the type of blast that you want to do the algorithm. So you click that particular blast. So if it's blast P, you click blast P. We have another set of blasts which I will talk about in the next slide. We have a PSI blast, which is a position specific iterated blast. And also we have phi blast, pattern hit initiated blast, So based on the work that you are doing, you now click on the right parameter and then you click submit. So the results depending on the size of your sequence, and then the results will be presented to you in short time. Okay, if you talk about a PSI blast, which is a psi blast, we call position specific iterative blast. You take the results of a normal blast and then align them and generate profile of concerned position, and use that profile to what, to generate weighting score for the next iteration. So if you do that, depending on the time, the iteration period that you have selected, in order for you to give you your result. Then we have a phi blast, which is a pattern hit initiated blast. So you have to provide a pattern alongside your protein. Your protein, maybe a kind of a motif or a kind of a expression pattern that you have gotten from somewhere. So you have to what, provided that. Then the database it have to contain this pattern, and then the similarity will be done with the rest of the sequence. So the result can be initiated also with phi blast as well. That is the result that you obtain from the phi blast. You can also use it to conduct your PSI blast. So what is the difference between blast and faster? Blast is very fast, good with proteins, produce good local alignment and short global alignment. It produce heat specific, HSP, heat specific protein reports, internal match in long sequence, and also might miss a potential alignment due to ruling out of sequence early in the process. Good at finding siblings. But what about faster? Not as fast as blast, very slow than blast, and much better than for with DNA than blast N. That is, can generate results for DNA than blast N. It produces Smith-Waterman's alignment. Local alignment, not global. Check each possible alignment with database sequence, and good at finding cousins. You can see finding distance relation, not your own immediate siblings. Somehow distant relation. Now, let's talk about multiple sequence alignment. MSA. Multiple sequence alignments are used to predict functionalities of different sequence alignments. You have more than two sequence from 3 to 1,000, from 3 sequence to thousands of sequence. So you want to predict the functionality of those sequence and see the area that is very functional within those set of sequence. Then it can be used as a step towards determining phylogenetic. That is, you want to construct a phylogenetic tree, you must first do your multiple sequence alignment. Then it can also be used for structural prediction, when you align one particular sequence against one particular structure against another structure to see the area, to see their root mean square deviation, and also their what's called RMF and the rest like that. Then you can use multiple sequence alignment of that particular structure. Multiple sequence alignment, multiple structural alignment, they goes together. Then for protein analysis, basically for protein analysis, but sometimes can be used for RNA analysis. RNAs has their own structure, so you can align the sequence of RNA and then the structure of RNA to determine their functionality. And also, based on the phylogeny that you constructed, it can help to distinguish between autology and paralog. That is, you can help us to find the autologs and paralogs. So ideally, it help to build to build a multiple cell alignment through weighted sum of pairs, but it is too computationally intensive. It is too computationally intensive. So as a result of that, it makes use of a heuristic and progressive alignment method. That is, this multiple sequence alignment can only work with a heuristic algorithm, blast and fast, and then the phi blast, and then the PHI blast. So those are the algorithms that can help us to do multiple sequence alignment in a good time. You don't have to wait for days. Okay. So one of the important tools that has been used even for multiple sequence alignment is cluster W. Cluster W it was first developed in 1988. 1988 started with cluster 1 to 4. Then we have cluster V in 1992. Then we have a cluster W in 1994 to 2005 with the variant cluster X. So these are the tools that has been developed to align sequence of a protein or sequence of DNAs together. If you want to know more about multiple sequence alignment tool, you can visit www.ebi.ac.uk. Then you see under the button the tab there, tools. So just click on the tools. Once you click on the tool, then go to sequence analysis. So we have different types of algorithm that you can use to conduct sequence analysis. So we have the align, the K-align, fit, Mozu, and T-Coffee, and the rest like that. So there are other resources that are available also on this particular site, which you can lay your hand upon. So for the clusters, we have different clusters that has been produced for multiple sequence alignment. So we have the cluster omega, which is the newest cluster variant that that was developed based on the E.D. Markovnikov profile techniques, HMM profile techniques. So we can see it can be used mainly for protein analysis. If you want to get the best result, it can be used mainly for protein analysis. That is just even the part of the disadvantage. So this particular cluster omega, you can use it to align very well protein sequence. Then we have the cluster W2, we have K and Lima, fit Mozu, T coffee, as I have said. And then the cluster W, this is how it works. Advantage that it's fast, not too demanding, widely used, fine for most uses. Then disadvantage is that fixing early alignment, that is, you can generate error. It doesn't search far, that is, it doesn't go far in the terms of comparing the area of similarities or differences. Then compresses gap. The gap scoring is not easily spelled out. So the cluster omega, which I said is an improved tool, you can access that www.ebi.ac.uk/tool/MSA. Then you have cluster omega. So it works based on it improved based on two areas. Number one, it generates a guided tree. There it creates a HMM profile alignment, E.D. Markovnikov model, which is a kind of a neural network to determine the optimal alignment of each of the sequence. So it helps even to what? It has a better speed and accuracy, and it can be work upon even on a different computers easily. Different computers easily. Let's move on. Okay. So another tool is coffee, the consistent base objective function for alignment evaluation. For alignment evaluation. So this particular coffee is a MSA tool and it's more accurate than cluster W and much less prone to problem in the early alignment stage, but it works very slow. It works very slow. So the variant of coffee is tea coffee, which is a tree-based coffee. Tree-based coffee. So it's also based on a heuristic approach and it's more faster than coffee, but it's still not as fast as cluster omega. So there are other tools available even on the site. We have Mozu, okay, Mozu. We have Mafat, MAFFT, and then we have K-align, K-align. So if you have your sequence and then you want to determine which particular tool you need to use, which particular algorithm do you need to make use of, if your sequence is between less than 100 sequence, then majorly all the major tools can help you to analyze, can make use of all these tools even to analyze it. But if your sequence is above 100 and but below 500, the sequence is below 500, then cluster omega, Mozu, and Mafat will help you even to do that very well. Then if it's greater than 500, if greater than 500, specifically for protein, cluster omega can handle that easily, and K-align can handle that easily. Can handle that easily. But when you have a small number of unusually long sequence, then cluster W and then K-align will help you in doing those analysis. So when you have your alignments, there is possibility of editing the alignments. Okay. So you can manually edit your sequence. Okay. And then you can visualize the data, you can manage the data, you can export and import the data based on the alignment that you have done. Yes, sometimes you have to look over even the alignment results and then input your own insight as a human being even into the results, or based on your area of interest. Okay, maybe you want to delete a particular amino acid from the sequence and see even their effect on the alignment. So you can edit your alignment manually. So there are some examples of a tools that you can use to edit your alignment. We have a cinema, we have GDE, we have GeneDoc, and we have Bioedit. So Bioedit, you have access to both the softwares within the Bioedit, and also it can link you up even with the online applications. It can link you up with online applications. When you have edited even your sequence, then you can, from your Bioedit software interface, you can send it to blast online when you connect to the internet, and then you generate your results, and then you continue with your work. Yes, we have talked so far even on sequence alignment. Now let's talk about how we talk about protein structure and function analysis. As we know that protein is a polymer that made up of different amino acid, as the monomer, and we have 20 natural or 20 common amino acid. I call it natural because they are the ones that you can find easily in most of the protein. There are some ones that are not common. We have like selenocysteine, SC. It's not common, but it's part of some protein, and we have some other unnatural amino acids that occur at some instances. But these 20 that you can see even on this screen, they are the ones that are natural and common amino acid, and they are classified based on their polarity and then based on their aromatic group, based on their non-polarity, based on their charge, negative and positive charge. So the positive charge can still substitute each other. That's what we call similarity. Okay. Negative can still substitute each other. That is called similarity. But when you have the polar may not be able to substitute non-polar, it will cause a kind of a devastation to the structure of that particular protein. So we can say amino acids are grouped based on their R group, based on their polarity, their tendency to interact with water at certain particular pH, and based on their based on their charge, as I have said. So one protein, one amino acid differs from another just at the R group, just at the R group, just at the R. So you have to understand the R group of amino acid in order for you to appreciate even protein structure and the protein function. Protein structure and protein function. Each of these R group, they have their different ionization states, and they have their own way of reactivity. Reactivity, that is, they undergo different reaction. Okay, and then there is a way that you can test based on the side. You can test for a particular protein based on the side group reaction. Cysteine side group, that is sulfhydride group, can only be detected or reacted with mercaptoethanol, Hellman's reagent, thiotritor, and some other chemicals like that. So you can detect, okay, if this particular protein contains sulfhydride group, and there are some reactions of amino acid, the one peculiar even to histidine. It means there's a group of histidine, and also the one for arginine, and then for the what is it called, a Sanger reagent, it can be used to determine the N terminals of amino acid. So you have to understand the reaction of amino acid in order for you to grab the concept of a protein structure and then protein function. So as I have said, you have to understand the amino acids, their three-letter name, their single letter name, So you have to be so sure, because proline and phenylalanine, they start with PP, but the single letter of proline is P, while the one of phenylalanine is F. For lysine and leucine has a single letter of L, while lysine has a single letter of K. Threonine and tryptophan and tyrosine. Threonine has a single letter of T, tyrosine has a single letter of Y, tryptophan has a single letter of W. Alanine and arginine. Alanine has a single letter of A, arginine has a single letter of R. So you have to take note of these three letter and the single letter of amino acids so that you can understand the protein sequence and also protein structure, and then ultimately protein function. So levels of protein structure, we have the 5 levels. We call it majorly 4 levels, but one of the levels are substructure. Okay, we have a primary structure of protein, which is a kind of a random fluidic arrangement of amino acid in a random fluidic arrangement of amino acid. Okay, when you have set of amino acid linked together by peptide bond only peptide bond. Yes, we call it linear, but in the real essence, it's not linear. Okay, it's like of fluidic, it's like of wave-like, because due to the side chain, the weight of the side chain will draw the configuration of the bond, and then it will determine how that particular amino acid will position in the medium. Then that is amino acid sequence. Then for secondary sequence, we have the formation of a backbone conformation. We have the formation of alpha helix, a kind of a ribbon-like. Then we have beta strand, and then we have the unstructured, which is a kind of a coil. Unstructured, which is kind of coil. Then we did this, what is it called now? The secondary structure, we have what is called super secondary structure, where the helix and the beta strand, they interact differently. So helix bundle with helix by hydrogen bond and the van der Waals and disulfide bond.
and then while beta strand form what we call beta sheet, different motif. There are different motif for supersecondary structure, and this motif serve as the basis of that protein family and then protein classification. So the supersecondary structure is very, very important. And then we have what we call tertiary structure, when the secondary structure, the supersecondary structure now fold to form a definite functional structure of that particular protein. If it's an enzyme, then the active site must be formed at the tertiary structure. Okay, so this particular tertiary structure is stabilized by the hydrogen bond, allostatic interaction, that is the charge of one amino acid interacting with another charge of amino acid, the opposite charge of amino acid. Then we have van der Waals interaction. Then we have a disulfide bond, cysteine linked with cysteine to form disulfide bridge. Then we have a shape complementarity. That is, the shape is well formed in such a way that if two polypeptides want to form a particular structure, they form a definite shape that complement to each other. Take for example, chymotrypsin, okay, or trypsin. So we have two or more polypeptide chain coming together to form a single structure, okay, as a sort of that post-translational modification. The unwanted part of the initial sequence has been cut off. Then the actual sequence that necessary for the formation of tertiary structure have to form what we call complementarity shape. So they have to group themselves together. The polar we arrange in a polar way, while the unpolar, non-polar side chain we arrange and bury inside. Then the formation of domain, as I have said, and then the formation of active site is formed at this particular point. Then we have what we call the quaternary structure. Quaternary structure is when you have two or more tertiary structure interacting together, and they are still stabilized by hydrogen bond, electrostatic interaction, van der Waals interaction, disulfide bond, and shape complementarity. The essence of quaternary structure is for their functionality. Most of the allosteric enzyme or allosteric protein, they exist at the quaternary structure. Take for example hemoglobin. Hemoglobin has 4 subunits, and each of the subunits is functional, can perform the function of a transportation of oxygen and carbon dioxide. And then you can see even there are some allosteric enzymes that has to regulate even the influx of a particular pathway. So allosteric enzyme also, they have this quaternary structure, big structure, oligomeric structure, so that their capacity to regulate and their capacity to perform a task that requires a high volume of substrate. Let me put it like that, high volume of substrate. And they are often present at the peculiar junctions in a pathway, and also peculiar functions even within the biological system. Talking about protein structure database. Protein structure, that is the three-dimensional structure of protein, experimentally from the wet lab, it can be derived by 3 methods. Number 1, by X-ray diffraction, or what we call X-ray crystallography. Then number 2, we have nuclear magnetic resonance, NMR. And then number 3, which is the latest one, is a cryo-electron microscopy. Okay, so these 3 methods can be used to resolve the protein structure in their 3D, XYZ. Okay, and then once this particular structure has been determined, it has to be curated into protein data bank. One example of protein data bank is a PDB, which is a www.rcsb.org. So all the publications that support the production of a particular protein structure will also be submitted together with that particular protein structure, so that anybody that wants to access that particular protein can understand the kind of organism that was used, even as an expression organism for that particular protein, the expression system for that particular protein, and then we're able to determine the resolution of the S-tray of the NMR that was used, the parameters that were used in determining the structure of this particular protein. And also from the site, you can download even the structure of that particular protein in a faster format. You can download the PDB of that particular protein. Then you can download the sequence that make that particular protein structure, which you can compare with the raw sequence. So by the time you compare with the raw sequence, you're able to see the areas that are not relevant for the formation of that protein structure. So the sequence of protein structure will different or sometimes differ from the primary structure in most cases, but not in all the cases. Okay, so you can download the structure from this particular site, rcsb.org. So from the site, as I have said, if there is existing ligand that was co-crystallized in the production of that particular protein, then you can see the information about that particular molecule, that particular compound will be also be spread out. And then the number of amino acid sequence that make up your protein will also be spread out, and then the organism in which it was expressed, it will also be spread out. If there is any mutation that was done on the protein, a kind of a recombination that was done even on that particular protein, the information about that also will be spread out. So talking about to visualize your protein, okay, there are different tools by which you can visualize your protein. We have RASMO, which is software, and then we have RASTOR, we can access online. Then we have Swiss PDB Viewer, which is online too. Then we have tools like PyMo, you can use to visualize your protein structure. You have a Chimera, UCSF Chimera, okay. Also, we have there are other tools that you can use to visualize even your protein. You can use to visualize your protein. Then for multiple structural alignment, that is, you want to align two or more structures together to see their areas of a deviation, root mean square deviation. Okay, how one particular structure deviates from another. Okay, you can use example of a site that you can do that is a DALI. Okay, help us to compare structures on the it's a web server that you can use to compare different structure. Then we have a combinatorial extension, which is a web program that also help us to determine or to compare structure based on the intramolecular distance. Then for us to determine different secondary structure prediction in a globular protein, okay, you can use PSI PRED, you can use SS Pro, you can use Procta, you can use Prof, you can use JPRED. That is for the globular protein. There are also other tools that you can use to determine transmembrane protein. So those ones, they have to consider the topology of membrane in which those particular protein are embedded. So to determine transmembrane protein is somehow harder than determining or predicting the second structure in globular protein. Okay, in globular protein. So you have to take note of the type of protein that you want to predict, and then make use of the right tool for you to do the prediction. So one important thing even in the protein structure and function is protein motif and domain prediction. If you talk about motif, it's a short segment of conserved sequence that is associated with distinct function in a particular protein or DNA. In DNA, you might have heard about palindrome. Okay, palindrome is a kind of a motif in a DNA sequence. Okay, a palindrome, you have a repeating pattern, A, followed by A, D, and you can read it in the complementary. They have the same in the complementary strand. Okay, when you have A in one strand, you know that the opposite strand will be T. Okay, so whatever be the pattern in a particular DNA or in a particular protein that have a distinct function, they refer to it as a motif. Those palindromic in DNA, that is the area where the restriction enzyme where they target. So each of the traditional enzymes, they have specific palindrome at which they what as their cleavage site, So okay, and that information and that particular insight has been has been advanced even to what we call CRISPR today, gene editing, okay? So you have to understand motif, and those are the segments of your DNA, of your protein, that have a distinct function. Then if you talk about domain is also a conserved sequence pattern, okay? And defined as the independent functional or structural unit. Independent structural or functional unit. Domain is somewhat bigger or longer than motif, okay? Somewhat bigger or longer than motif. It can consist of up to 40 residue, between 40 residue and up to 700 residue, okay, depending on the specific gene or protein that you are considering, okay. So the two are evolutionary more conserved, okay. Domain, motif are evolutionary more conserved. So that means those areas serve as a specific usefulness, as specific function in that particular protein, that if anything tamper with it, that particular protein will lose its function, will lose its identity. Okay, that particular gene will lose its function, will lose its identity. So domains and motifs are evolutionary more conserved than other regions of the protein. Okay, so for you to determine protein motif or gene motif, there are sites that you can do that. Example of those sites are listed even on the slide. You have the pro sites, e-motif, prints, blocks, pfarm, and then interpro. Okay, interpro. Okay, this is a kind of a site where you can do your motif scan, deposit your protein there, protein sequence, and then you click the relevant parameter, and then you submit. So it will give you a kind of a motif, a signature of that particular protein, that is signature that is peculiar to that protein family, okay. For this particular motif scan, it will make use of a ProSite, PFAM, and Interpro. So it will work using the three together by clicking the results that you want to generate. And also for the Interpro, which you can access by the use of EBI site, www.ebi.ac.uk/Interpro. There also you put your sequence in faster format, and then you search, you'll be able to see the motif, the pattern that your protein has. We have talked about protein secondary structure prediction. Then we have to determine the protein tertiary structure. Until your protein assume a tertiary structure, your protein is not functional. Until your protein assume the tertiary structure, at least the tertiary structure, it can go to the quaternary structure, but until it gets to the tertiary structure, it is non-functional. So this one helps us or lead us to determine or predict the tertiary structure of our protein. Protein structure prediction aims to reduce the sequence structure gap. As I have told you, there are a lot of sequence that has been generated from DNA, and then the computer program like a BLAST can help you to assess or to predict the corresponding protein sequence. But protein sequence doesn't give us specific functional insights. So we have to find a way of deduce the structure of those protein. So experimentally, it is time consuming, it is costly to use all those three methodology, X-ray diffraction, NMR, and cryo-electron microscope to determine a particular protein. So in order to bridge the gap, then we have to predict even the protein structure based on the existing knowledge of those experimentally determined structure. Okay, so the structure determines function. As I have said, structure determines function, and until your protein assume the tertiary structure, it is not considered to be functional. So there are many important proteins which their sequence are available, but their structure are not known. There are so many proteins, even in various organisms, including human beings, that we don't know their function up to today. And they are there until research gets to them, then we're able to understand their role that they play even in their normal physiology or in the pathologic arena. Then, as I have said, we have different methods by which we can model or we can predict our protein structure. There are three major computational approaches that has been established to determine the three-dimensional structure of your protein. Number one, homology modeling. So this one builds an atomic model based on an experimentally determined structure that is closely related at the sequence level. That is, the template that is being used for homology modeling is a kind of experimentally resolved protein. That is, the structure that has been obtained from the wet lab, okay, is being used as a model to build or to predict the structure for your own protein. Then we have another one which is trending. This one identifies proteins that are structurally similar with or without detectable sequence similarity. Based on the detectable sequence similarity, and then we have ab initio approach, which is a kind of a simulation based and predict structure based on physicochemical principle governing protein folding without the use of a structural template. Ab initio make use of the principle of protein biophysics, in which the one that fold best will have a lowest gives free energy. Lowest gives free energy. So based on the folding pattern, the one that has the folding that has the lowest give free energy is said to be the best even structure. It's based on the ab initio model. Okay. So homology modeling, as I have said, you have experimentally determined structure which you use as a template. Then, based on that particular template, you develop a model. You develop a model structure. Model structure, okay. So AlphaFold, which is a kind of computationally predicted structure, has been made available on UniProt for most of the protein. For most of the protein, it makes use of a deep learning combined with all the artificial intelligence to predict different structures from sequence. Okay. So you have to what you have to, you can even read more about that. Okay. So ab initio is more of simulation. You have a fold at home, you have Rosetta. Those ones are based on the ab initio model. Like trending, we have a Raptor X, we have Fires 2. So those ones are based on trending. But if you talk about homology modeling, we have some homology modeling softwares and server, like a Swiss model is based on homology modeling. So all these ones, they help us even to what? They help us even to predict even our protein tertiary structure. So homology modeling predict protein structure based on a sequence homology with the known structure, standard structure, and it produce all atom model based on alignment with the template. Okay. To do homology modeling, we have 6 steps. You have to consider 6 steps. Number one, you have to select a good template selection. Okay, it is based on the percentage identity, percentage similarity, and then the types of tertiary traits. Is it hetero? What's it called now? Is it monomeric? Okay, or is it polymeric? Okay, so if it exists as one subunit or as two or more subunits, so your template will determine what you will predict, will determine the result of your prediction. So choosing the right template is very, very important in homology modeling. Then number two, alignment of the target and the template sequence. Okay, the alignment will be done between your own sequence and then in alignment with the sequence of that particular template structure. Then number three, the homology algorithm will build framework structure on the target protein consisting of the main chain atom. Then number four, model building uh, include also addition and optimization of side chain loop and then atoms. So in order to prevent what we call clashing, okay, it has to what? It has to refine the model in such a way that the amino acid side chain, they are not what? They are not clashing with each other. So it has to refine the model and then ensure that your own sequence give a model that has the right loop and then the right atomic arrangement of your side chain. Then number 5, we have a refinement and optimization of the entire model according to the energy criteria. So this particular model, we still find the model that give the lowest energy criteria. Okay, the lowest energy criteria, like switch model, it has the energy criteria that is what a mean. Okay, so that one try to what? To evaluate based on the Ramachandra plot to see the area that is allowable and the area that is allowed. So if you look for the energy criteria in terms of a gift free energy to see, okay, how the models constructed, how they perform better. Then number 6, evaluation of the overall quality of the model. Evaluation of overall quality of the model. Okay, so the alignment and the building will be repeated, iterated until satisfaction. So you can see the kind of a figure even on the screen show you the steps involved even in the homology modeling. In the homology modeling, you have the target sequence, which is you use it to search the database. Then, based on the identities and similarities results, you choose the best template, and then the best template we try to align again. You refine the alignment, then we build a model framework, and then the loops and the side chain are what are properly set to avoid clashing and what we call steric hindrance. And then after that, the model will be optimized, and then the optimal model, the model that have the best optimization in terms of energy criteria and in terms of a comparison with the experimental parameters will be selected. Okay, so these are the sites. We have 2 sites here for homology modeling. We have a switch model, and then we have a Fares 2. Now let's talk about molecular phylogenetics. Phylogenetics is the study of evolutionary history of living organisms using tree-like diagram to represent their family history. Okay, so the tree pattern referred to as phylogeny. So the aim of constructing phylogenetic tree is to determine the evolutionary divergence or convergence of your sequence. Okay, nucleic acid or protein sequence can be used for your phylogenetic construction, phylogenetic tree analysis. So molecular phylogenetics allow the suggestion of phylogenetic tree for a given set of sequence. For you to construct phylogenetic tree, you must have more than two sequence. Because phylogenetic tree has to be built based on multiple sequence alignment. So for you to construct a good phylogenetic tree, you must have more than two sequence, either DNA sequence, nucleotide sequence, RNA sequence, DNA sequence, or protein sequence. So in the construction of tree, there are some terminologies that are involved that you have to understand, okay? So when you have a tree, the initial point where that particular tree started from is called the root. That is where the ancestor is. That is where the ancestor is located. So you call the root. So, and then the root, as it forms even a kind of a branches, okay, we have a kind of a branches just like a tree. You have a branches from the stem, you have a branches, and then the point where the branches also give birth even to another branch, then we call that internal node. So the tip of the end part of each of the branch is called tarsal. So when a particular branch divided into two to form a two segment, we call that particular point dichotomy. But when it gives back to three or more branch from a particular single branch, then we call that polytomy. Okay, so a node, is a point that connects two adjacent branches. Okay, a node is a point that connects two adjacent branches, as you can see in the figure above. Then a branch pattern depicting the ancestor-descendant relationship on a tree is called lineage. When you can trace from the root, you trace to the tarsal, then you are called that particular pattern, that particular part is called lineage. That particular is called lineage. And then that's what we call the branching pattern. The branching pattern of a tree is called tree topology. As I have said, you have dichotomy and then you have polytomy. Okay, you have dichotomy and you have polytomy. And then tree, the can either be rooted or unrooted. So rooted is when you define that particular relationship based on a particular ancestor. That is, you specify that, okay, this particular tree supposed or assumed to have a particular ancestor. Okay, so the particular ancestor may not be able, you may not be able to define it based on analysis, but you assume that what this particular tree has a particular root. For the unrooted, you construct your tree with a hope of what? Of getting the relationship between your tarsal without having definite root, without having definite ancestor. You want to see interconnection of just the tarsal without their comparison or without their relativity to the what? To the root. Okay, and then we have different form of root, different form of tree presentation. Different form of tree presentation. We have cladogram and we have phylogram. Cladogram is when your tarsal are arranged even in a unique form, in a similar platform, okay, without giving even a relationship between the distance even from the root, without considering even the actual distance from the root. But for the phylogram is a form of a tree presentation in which that the tarsal have a specific distance from the root. So the distance from the root are established in the phylogram trees presentation. Phylogram trees presentation. So this will help even when you want to do your microbiota even study or metagenomics phylogenetic tree construction. You'll be able to see the closeness even of your microorganism or the closeness of your plants species even from one another. So procedure for constructing phylogenetic tree. So there are procedures that you have to follow for you to construct phylogenetic tree properly. There are 5 steps. Number 1, you have to choose the right molecular marker. Is it the DNA sequence that you want to work with or the protein sequence? I have to tell you this. Molecular phylogenetics is different from normal biological phylogenetics or fossil phylogenetics. Molecular phylogenetics make use of either your DNA, your RNA, or your protein sequence. This time around, we are looking at the molecular composition, either a specific gene or a specific enzyme. You want to what? You want to see their evolutionary trend. So this one is different from the biological phylogenetics, where you consider the evolution of a particular species. For you to do that, even in terms of molecular, you have to consider all the genomic content. That is the genome constituent even of that particular organism against the total genomic constituent even of another organism. That's why you can establish even their species evolution, Okay, and even beyond that, that's what we call epigenetics. That is, there are some genes or that has been altered as a result of adaptation to the environment. Okay, environmental factor has influenced even the methylation of some aspect of a gene. Okay, which you will not be able to trace. But what molecular phylogenesis is all about is that you choose a particular gene or a particular protein or enzyme, and you consider its evolutionary trend with another set of that protein in another organism. Okay, in another organism. So you choose the right molecular marker, DNA or protein sequence. Then number two, you perform multiple sequence alignment. Multiple sequence alignment. That is, you align all the sequence that you want to construct phylogenetic tree upon. You align them together so that the algorithm will be able to see the area of conservation and the area of their divergence, even from the result of a multiple sequence alignment. And also number three, you choose a model of evolution. A model of evolution. For the nucleotides, the nucleotide substitution model like Jucanton and the Chimera models can be used. Those are the two major models if you are working with a DNA sequence. Then for the amino acid sequence, you have to use amino acid substitution model such as PAM. I've mentioned PAM the other time. Okay, and then we have Jones, Taylor, Thornton, JTT model. JTT model, that is another beautiful amino acid substitution model. Amino acid substitution model. Then number four, we have you have to determine the tree building method. The tree building method. So two methods can be used. Number one, you can use even distance-based method, or you can use character-based method. Okay, distance-based method also can be divided even into two. Number one, we have what we call clustering-based method, such as unweighted peer group method using arithmetic average, UPGMA, UPGMA. Then we have what we call neighbor joining, neighbor joining, neighbor joining. So these clustering-based methods are very fast, but they may not be accurate. They are very fast, but they may not be accurate. Then the second part of a distant method that we can use is a optimality-based method. Optimality-based method. So example of such is a fit, Margulies, and also minimum evolution method. So these two methods, also the optimal-based method, they are very fast, they are very accurate, but they are not fast. They are very accurate, they are not fast. They are not fast. So the second method is the character-based approach. Character-based approach. So this method, you can either use maximum parsimony and the maximum likelihood method. Likelihood method. If you want to read more resources, I will point you to the resources which you can use to read more about molecular phylogenesis. Then lastly, number five, you have to assess the tree reliability. The tree reliability. So not that you have constructed a tree and then that particular tree is as authentic as it's supposed to be. No, no. So the method that you use, the approach that you use will determine the type of tree that you constructed. So you have to assess the tree reliability by subjecting it to iterative what's it called, assessment, iterative assessment, to see if after you construct different models of that particular tree like 100 times, you still get the same tree pattern. You still get the same tree pattern. Okay, for you to do that, there is a particular method that you can use to do that, which I will explain later. So this is how the tree look like. The one you see with blue is a kind of circular presentation of a phylogenetic tree. Then the one you see even below is the one that is a kind of a linear presentation of a phylogenetic tree. You can see a number there show a kind of the distance of which a particular taxa is far or have evolved even from the root. Then we have multiple sequence alignment of DNA sequence. That is how it looks like. You can see a segment with star. Those are the areas that are conserved. With star, those are the areas that are conserved. You can see G, 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 C, 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 C. Then you can see the gap. Therefore, the protein multiple sequence alignment. Okay, if you work with a cluster O, then from the cluster O page, you can generate the phylogenetic tree. You can see the area of star, the area of star. Those are the areas that are absolutely conserved. Absolutely conserved. You have F, 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 F. The area with two dots, they are somewhat conserved. You can see just then you have IV at the beginning. But the area with single dots, they are partially conserved, somewhat conserved. Okay, then the area where you have gap, those are the area that they are not conserved at all. So once you have done your multiple sequence alignment, then you can construct your phylogenetic tree. Look at the figure even on the right hand side. You can see from the early globin gene, you have a gene duplication, alpha gene and then beta gene. So from the alpha gene, you can have the alpha gene in different species of animal. The frog, which is an amphibian, you have a chicken, which is a bird, then you have mouse, which is a mammal. So you have both the alpha and beta, and then for you to now determine what we call the sequence interpretation, biological interpretation, then you have to look at it in form of a relatedness. Okay, so all those alpha in frog, chicken, and mouse, they are referred to as the autolog. When you have genes, same gene in different species performing the same function, then you have what we call the autolog. It occurs as a result of a speciation event during the course of evolution. Then for the paralog, that is within the mouse, you have alpha and beta. That one is a paralog. So within the same species, you have two genes or two proteins that perform usually different function. The way alpha will behave will different from the way beta will behave. So as a result of that, we refer to it as a paralog. So you can see some definitions over here. Homolog is the one combination of both that is based on their similarity. Then within the similarity, you can now distinguish, okay, those that occur as a sort of a speciation event, or which is that is an autolog. Then those that occur as a sort of a gene duplication, which is a paralog. Then we have xenolog, which is a result of what we call lateral gene transfer. Lateral gene transfer between microbes, beneficial microbe, and then let's say and the host, a kind of a symbiosis relationship. Okay. So as a result of that, that's what we call lateral gene transfer. Okay. So how to assess your what's it called? The reliability of your tree. There's a particular method that you can use which is called bootstrapping. It's a statistical technique that can use to sample errors of a particular phylogenetic tree. So it will do the tree, it will reconstruct the tree in different formats over time, and see if you still get a particular tree eventually. So once you can get a particular tree after a lot of trying, then you can be sure that okay, that particular tree is the actual one that can represent even the course of evolution for that particular gene or particular protein. We have different tools that you can use even to construct a phylogenetic tree. You have Mega, you have Phylip, you have POUP, you have TreeView, you have Foden, you have PhyloWin, you have Tricon, you have Dredon. As I have said, from the cluster O on EBI, you can construct even your phylogenetic tree. Also, from the phyllo O, you can even copy your tree data. That is a tree, phylogenetic tree data, you can copy it and go and visualize it in a high definition manner on a phyllo and then also on iTor web server. So what are the applications of phylogenetic tree? Number one, it can help us to easily determine gene duplication. That is when there is error in duplication of genetic information, which can result in either deletion or insertion of a particular nucleotide and then particularly amino acid eventually, which can affect function even of that particular protein, the way it's supposed to behave. So as a result of that, you can use phylogenetic tree to determine that. Then lateral gene transfer, as I have said, as a sort of a symbiosis or a kind of a dysbiosis. This is what we call the transfer of gene from a coexisting microbe together with the host. Then recombination during meiosis. Phylogenetic can help us to determine that how a particular trait has survived the course of evolution. The course of evolution. Then number 4, mutation in splicing pattern of introns can affect exon function due to positional shifting. Okay, when introns, which are the non-coding parts even of the genome, when the splicing during the recombination process occur, then positional shifting of the intron can affect the position of the exon. As a result of that, can affect even the function of the coded gene product. Then number 5, genome-wide association study. Mutations in a gene that are passed on from the offspring and then coexist with the original gene will result in what we call polymorphism. Single nucleotide polymorphism is when you have a single nucleotide changes in a particular gene, single nucleotide change in a particular gene, and that can be what can be assessed by genome-wide association study (GWAS) by using the polygenic risk score, and then you use all the other genomic assessment association study to assess even the single nucleotide polymorphism in that particular gene. Then we have what we call phylogenetic tree can be used to identify microbes and then help in their classification. Those that do the metagenomics, they have to take in even the ribosomal DNA or ribosomal RNA of that particular cultural microbe, and then after they have sequenced, they get the sequence of that ribosomal DNA or ribosomal RNA. Then they do the sequence alignment using BLAST to blast it to identify a particular microorganism sequence on the database that match that particular their query sequence. So as a result of that, they can identify the types of microorganism that is present even in their culture sample, and then they'll be able to classify the function of that particular microbe even from those analyses. Yes, having said all this as an introduction even to bioinformatics, so there are resources that you can access even online, textbooks that you can access online in order for you to read and prepare yourself even for this field of bioinformatics. So I have listed a few of those textbooks even in this particular slide. The one that you have to even lay your hand upon, which is easily freely accessible, is Essential Bioinformatics. Essential Bioinformatics, written by Jin Zayong, a professor in Texas A&M University. And then we have Basic Applied Bioinformatics, which is a very good textbook. And then we have a different volume produced by Springer, Essential of Bioinformatics, Volume 1, Volume 2, Volume 3. And then there is this particular textbook on phylogenetics, which you can easily access in order for you to study more about phylogenetics. And then there is this a kind of a manual by SRM University, which you can lay your hand upon to start even the step-by-step and then follow how to conduct basic bioinformatics analysis. And then there is this agricultural bioinformatics for those even in agricultural field. Bioinformatics cut across every aspect of living thing, animal, plants, microorganisms. So no matter be your field, you are studying animal, biomedical, veterinary, pharmacology, anything that deals with life, you have to understand and develop the skill of bioinformatics. Then there is this particular one that's linked to drug discovery, knowledge discovery in bioinformatics, and then the other one was published by Wiley. Then we have bioinformatics for vaccinology, and then we have RNA bioinformatics. Okay, there are a lot to be done in the times of RNA bioinformatics. We have different types of RNA, tRNA, mRNA, microRNA, and short coding, non-coding RNA. So all those areas of RNA still need to be explored. So using this particular textbook, it will open your eyes even to those particular fields. Then we have bioinformatics for glycobiology and glycomics. Glycobiology is the study of carbohydrates. Yes, we don't need to limit ourselves even to DNA. We don't need to limit ourselves even to RNA or to protein. We have to explore even the areas of glycomics, that is the carbohydrate, lipidomics in the areas of a lipid, because we have the interaction between these. We have a lipoprotein, we have glycoprotein. So these particular resources will open your eye even to how bioinformatics can be used to explore glycobiology and glycom. There are a lot of resources that you can lay your hands upon to develop yourself even in the areas of bioinformatics, specifically applied bioinformatics. Okay, applications of bioinformatics. Bioinformatics has been used in various fields, as I have said, in the area of microbiology, molecular medicine, personalized medicine, drug discovery, evolutionary study, environmental study, biotechnology, climate study, crop improvement, forensic analysis, and also in terms of development of insecticide, and then improvement of nutritional status of nutritional quality in food, that is genetic modify organism, GMO, strain improvements, veterinary to cure diseases related to the animals, and then to those of plants in terms of drought resistant varieties development. Now, let me just show you some of the work that I have done, even in terms of bioinformatics, in considering the area of enzymology, computational enzymology, or enzyme biophysics. I have worked on effect of pH, on structural dynamics of HMB-CoA reductase, which is one of the key enzymes in cholesterol pathway, and then study the binding affinity of beta cystosterol to that particular enzyme, using bioinformatics. Also, I have worked on the area of bioactive peptide. I have worked on the area of bioactive peptide as a therapeutic for neurodegenerative disease using bioinformatics. Also, I have worked on the area of medicinal plants as an alternative medicine to solve biomedical disease. This work was focused on coronavirus, how to develop and how to find the cure even for the treatments of coronavirus disease, COVID, using 9 medicinal plants. Also, I've worked in the area of food, that is, bioinformatics can be used to assess the pharmacological potential of a food bioactive, that is, food grains that are plants that are of economic benefit. So you can use bioinformatics to assess the bioactive, predict even what those bioactive compound will do to the body when we consume the food. And also you have to understand the area of pharmacokinetics, ADME, that is the absorption, distribution, metabolism, excretion. And then the T, which is part of the ADME, is toxicology. So you can determine different parameters even of your food contents. Then I have worked the area of cosmetic development of natural cosmetics, that is the active ingredients that are natural, that will not lead to the formation of some diseases like cancer, like rashes, and the rest like that. So using the natural source, I've been able to work even on different on the use of medicinal plants as an alternative source in cosmetics development. And also I've worked the area of microorganism, that is the treatment of diarrhea, finding treatment, a kind of a target, a kind of a broad spectrum a compound that can inhibit even some of the targets, and that is the proteins, the enzyme in organisms that are causing diarrhea. Also, I work in the area of environmental science to determine the toxicological assessment of some toxic compounds that are often present in a contaminated soil and dump sites that can affect human health. Okay, so if you have question and answer, you can send the question to me, and then I'll be able to answer you. And also you can access these particular materials even from my ResearchGate profile, which you can download this PDF eventually. So if you have questions, send it directly to my email. I'll be glad even to provide answer even to your questions. Thank you.